kind of useful one for the FTR data analysis, so which he will talk about later. So we have personally invited him to join this panel discussion since there was no other opportunity for him to share this new software development. So definitely it will add value to some of the techniques that we have listened since morning. That sharing we will do it separately. First, let us start with some open discussion. Okay, I think this microplastic is a topic that has been out there for more than a decade, and people have started in different way. And now I just want to ask everybody how, maybe some of you have started the research only very recently, but Dr. Barbara or Othani, they are in this for almost 10 years. How they have seen this topic is evolved? Because for me as a user, if I look back some of the previous publications, they were just trying to look for the particles. Okay, so and sometimes there are false positive, sometimes false negative, and a lot of reports are basically on the number of particles. And again, the size segregation or the size classification were not really harmonized. But now, based on the real initiative coming from the European side, so everybody at least has a common understanding of the harmonization on the classification of these particles. So to address this microplastic analysis, how the technology has evolved and what were the challenges 10 years before and what we have and what we see as a development in the future research. So everybody can take 2 minutes to talk about their view on this. So maybe I request Dr. Barbara to start. Othani, and then we can go by this name.
Okay, thank you very much. So I think after all that time, we are continuously learning. And in the beginning, I think we were very creative and just have a good idea. But by now, we see more and more difficulties. And I think one of the biggest point, and that is something I try to point out in my presentation as well, that these particles are a real challenge, and we have always embedded them in a natural matrix if we're looking for environmental samples. So we have a huge discrepancy between pristine polymers and at least alterated polymers. And we need, on one hand, a comparison basis and an analytical basis to compare data. On the other hand, a good reliability. So we need a pragmatic approach, but also a continuous critical review of our own data, and to try, as I showed you, with blanks and whatever, to really show the real data we have and then discuss them. So I think the biggest effort is that from a political side, an enormous pressure is there on ideal analytical methods and ideal data, but I think there are still a lot of leakage in knowledge with respect to precise data. And I think from the thermal analytical point of view, we need a level of abstraction to discuss values we are generating.
Thank you. Prof. Othani, so please share your views.
Yeah. I have been analyzed industrial plastics by using the analytical method for a long time. But recently, we have started to analyze the microplastic. But it is very difficult to analyze, especially for the environmental real samples. And it's also very difficult to analyze smaller particles in the level of nanoplastics. So I think not real information is still not obtained for the nanoplastic. So the analytical pyrolysis is a possible approach to detect nanoplastics. But I think various problems still exist. For example, the effect of the matrix products and Another problem is their microplastics, origin of their microplastics is unknown. So it is very difficult to characterize them. So we need still more challenges for this analysis.
Okay, thank you. So, Lara, please share your thoughts on this.
Yeah, sure. So I've been working for Marks now for a couple of years using thermal desorption. So it's been a really interesting application area for me to learn how to apply this to different sample types, and particularly microplastics. And there's obviously a lot of different techniques, which I think is really interesting, and being able to use them complementary. I think for us in particular, being able to analyze the microplastic samples from a variety of different sample types at lower temperatures gives us the option to look at the full picture, I guess, and more environmental samples, and being able to see more of the other VOCs as well as marker compounds from microplastics, and being able to identify things like additives and source profiling and toxicity and things like that. And then also being able to turn those into concentrations that we can then look at and compare to existing standard methods and regulations for different environmental purposes, and toxicity and things like that from different government guidelines. So yeah.
Thank you. Yeah. So I think Dr. Siba, I saw him online. Okay, he's back. Okay.
Yeah.
So, yeah, share your thoughts and experience, because now we are switching from thermal analytical technique to the spectroscopic technique. So you'll be sharing views from the FAR side. Then we have Dr. Mike, and also
all right, thank you. So yeah, I have been into the topic for 2-3 years now, and it was quite the learning experience. So I wasn't aware that particles or the problem was already known for such a long time, and now really just has appeared in the public discussion. And yeah, regarding the particle detection and so on, in the short time I've been here, there have been drastical changes, starting from the idea to do everything with ATR and finally arriving at FTIR imaging for the solution, at least for the microplastics. And we are getting more and more inquiries about nanoplastics as well. And this is a topic where we're not sure at the moment what is the best way to go. There is still a lot to be discovered. Also, how important this actually is, potentially. It might be just as important as microplastics, but we don't know about this yet. And yeah, I'm very interested to see what's coming there.
Okay, thank you. Dr. Mike, can you share about your new technology? Because this technology is something considered new for many people. So
yeah, I certainly can feel the excitement. For me, I have started working, maybe my first encounter about microplastic analysis was maybe a few years ago. And at that moment in time, I can tell you that the sample preparation was, I would say, not sufficient to be able to, let's say, even just to, find the fragments, because there was just so much other stuff in that stew of material. But then I think, what I have seen in the past couple of years is that the sample prep has been gotten better, the digests are doing better without damaging the microplastics. I think that's very helpful because we just want to analyze the microplastics. With that front, I think when we analyze microplastics, we don't need the other stuff, like humic acid and all the other stuff. So just focus on the microplastics. Now, with that said, what that means is that once we have the good sample prep, then it gives us a best chance. So I don't think that there's something that we can skip out on. Now, the next thing is that now we have the microplastics, then we need to get a good, accurate analysis on these, whether it's large, small, nanoplastics or microplastics. We need to have a good identification of these microplastic fragments. Unfortunately, again, it's a very difficult problem because with conventional techniques, depending which would, like the spectral band shapes would depend on, the surface morphology, the diameter of the particle. They all come into play with the spectra that we see, but then they are not really changing the chemistry per se. So what we need in a technology is that we would be able to, okay, we got this particle here, irrespective of size and shapes, we can still get the actual IR spectrum that is representative of the chemistry of the microplastics. I think that's where it's going to allow us to figure out, are we really measuring the right chemistry here? And once we do that, can we build a good case and good toxicology study on how these particles are affecting the real biota? I think that is the important part. Once we have that, then once we have the high sensitivity, like what we have here with the Mirage OPR system, where we can measure 500 nanometer particle or even less, then we now have these analytical data where we can correlate with, let's say, a control study on a biological matter, and then build the statistics on, okay, how toxic, what is the toxicity of these microplastics and nanoplastics? And we now have the tool for doing so. I have heard from the talks that, okay, small particles are 10 microns, but when I was still a student, we were talking about how nanotechnology and nanoparticles are all toxic. That's when I think when people start paying attention, and then I think now I think we are coming to a point where we start to have the technology that would approach that point. Mirage is part to start, but I think it's a very good start for getting a good sense of what the chemistry or the real chemistry is in the microplastics.
Okay, thank you.
We look forward to this type of development where we actually get the good data, which will help.
So, yeah, thanks, Mike. So can I request Bentik, so you can share your research experience, also briefly tell us about the software that is used for based on my machine learning. Yeah. Okay.
Should I do the presentation or just commenting on the?
I think if the number of slides are not more, is okay. So you can keep something like a summary slide, so which people can see and listen. Yeah.
Okay, just a second. I thought it would run differently. Okay. Can everybody see my screen now?
Yes. Yeah. You do that.
Okay. So. Okay, thank you very much. So my name is Benedikt Hufnagel. I'm co-founder of a company called Purency. We're situated in Austria, and we focus on the data analysis of microplastics using machine learning. And regarding the question that was asked previously, I would answer the following things. So in my view, the current or the recent developments and challenges in the fields of microplastics as we witness them is a key topic is the robust spectral data analysis. So in spectroscopy, we have to deal with various kinds of matrices. We have to deal with polymer aging, the physical effects that we have in the measurement, for example, total absorption, scattering. If you use Raman, you have fluorescence effect. And of course, additives such as pigments and so on. We have issues regarding the comparability of results. So when using spectral data analysis software, we have a rich variety of parameters that we have to choose. And in this way, the results might not be comparable. And also we have different devices. And there is also a problem regarding the scalability of measurements. So in order to be able to provide microplastics analysis as a service, we need a high speed and we need to reduce costs so that this measurement becomes broadly accessible to a large community of people and industries. And of course, we have chances and limitations regarding different spectroscopic techniques. So in my view, these are the main challenges that we are currently facing. And in order to allow people to use this, we use machine learning as an alternative approach. And to give you an idea how this helps in this aspect, I have compiled a short study regarding different matrices that you can see here. So here, for example, by using FTR imaging in combination with a machine learning approach, we are able to detect microplastics in various kinds of matrices very robustly and in a very fast manner. So in less than 10 minutes, Just to give you an example, here we have plankton. These are samples from the literature. We have wastewater treatment plant samples, sediment, soil, compost, and also a sea salt sample provided by Bruker Optik. And we also have to make sure that data is comparable across devices. So, for example, the last examples that I showed you here were from Bruker Hyperion 3000 and Bruker Lumos 2. But we also have to address other devices, for example, Agilent Carry, Perkin-Elmer, or Thermo Fisher, and make this data comparable in a way. And last but not least, we have to deal with different kinds of samples and also very difficult samples in the future. So in this example, I show you a sample of a filter, which is almost completely full of sediment, but irrespective of that, machine learning can still detect the particles within the image. And this technology is not limited to FTIR, so it can be applied to various kinds of spectroscopic techniques. This is also the reason why we extended it with an initial prototype to also address Raman. And in Raman, of course, we have the issue of fluorescence and the different lasers which are applied. So in this way, we also want to create comparability between the different kinds of data. So thank you. This was just a short introduction about Purency and my background.
Thank you. I think definitely that the people who are working on spectroscopic technique will be really curious to see how your software can really solve their data analysis problem. And that's a good thing. Okay, actually, yeah, as Dr. Barbara was mentioning, so the legislation or the regulation people are looking for like some kind of information that will allow them to really come up with some kind of new regulation or law. Okay, if you look at some of the other contaminants, okay, for example, microorganic contaminants, okay, like polyaromatic hydrocarbons or organochlorine pesticides, they always come up with a kind of two threshold. One is the summation of the 16 pH, for example. Then they also have some specific threshold for certain which are considered more toxic or carcinogenic. So how do you see the legislation for microplastic will evolve? At least my when I was talking to few people, I was under the impression, okay, we may have similar like a threshold. There may be a threshold for microplastic as a total, just a composite sample where the summation of all microplastic should not be more than certain number of ppm. Then some of the polymers, like they are more toxic, then there may be some individual threshold limit. So this is what, that means it's more like a mass-based threshold limit is what it may come as a legislation. But even to reach there, we have to do some kind of this toxicological aspect, which we will be hearing from other researchers on day 3, where they have to look at the number of particles, size, shape. So definitely to assess the impact, it is a combination. But when it comes to as a kind of a limit or to control the pollution, what will be the strategy that the regulators will be looking for? Maybe I request one from the thermal analysis side, one from the spectroscopic side to give your feedback. What to look for as a new regulation?
Well, I think that currently
I'm offering also to give an answer.
Sure. Okay.
Okay, so currently I
Sorry,
think,
but please continue with thermoanalytical first.
Thermal. Okay, go ahead. Yep.
Yeah. Okay. Maybe Dr. Barbara, can I request you to share your thoughts?
So I think it's a very difficult question because so far non-reliable data on toxicology are there. And therefore it is a very difficult question. So for example, some regulations try to focus on a development. So saying we need the state of the art, and then we hardly appreciate or want to reduce a concentration or reduce a signal. And I think that is the best way in the moment to actively perform something with respect on this time pressure and societal pressure on this whole topic, because the toxicological projects are already running and they are presenting new results, but I think we are still far away from any precise toxicological statement. So I think the best way is complementary using both methods, and first defining levels that should be level down, and with new results, change these levels, adapt these levels to realistical parameters that are easy to follow globally, because that is another problem, because the equipments are very, very different.
Yeah, that's correct. Yeah. So thank you. Yeah, I think next 5 years, as you said, people will be looking at the development of state of art for regular monitoring. Then worldwide, people will start, because, for example, Singapore has a project, to look at the microplastics for 2 years. Then that data will be the base for them to discuss the next level of planning or anything. So similarly, something. Yeah, I think Bentik, you want to say something first?
Yeah, something to add to what Barbara Scholz-Böttcher said. I think there is also, we have to differentiate between science and standardization and legislation, because currently in standardization, where we also participate, for example, in ISO at 2 working groups, there is a lot of parallel work with standards and drafts moving into different directions, using different concepts for defining microplastic. And this is also true for the developments in the USA by ASTM International, and also regarding the California Senate bills. So I think first, before we can really set thresholds and do legislation, we have to agree on some common standards. But the standards that are now being developed, I think they are not really comparable and sometimes also contradicting each other. So this is an issue we need to solve first.
Definitely, yes. That's a contribution from the scientists. Yeah, Machan, what's your feedback?
Yeah, it's basically the same experience I have. So since legislation is still a long way to go, different institutions or different groups of industries are trying to implement their own solutions and come up with very different opinions on how to treat that, what methods are there. And yeah, well, there needs to be a common ground first before any kind of standardization can be done. I mean, we're trying to, put forward our opinion on the matter. But it's also only one of many, and there we need to find a common ground first.
Yeah. Like Laura, Mike, do you want to add any here?
Yeah, more like a thought. So currently, let's say when we test plastics for food contact or medical use, let's say EU, they do have this, let's say 10 milligram per milliliter, that type of limits. Now, when they extract the whatever that comes out, or like the migration test, they don't really qualify or like quantify what's in that 10 milligram. So perhaps one of the starting point could be, well, what is in that 10 milligram? Like, is that material in that 10 milligram enough to be toxic to people? Or like, how is that effect in terms of toxicity and whatnot? Now, again, this is on the testing side of things. And so far, I have not seen anything that is for microplastics for real. I mean, we do have something like that in the US where, like, California Water Board would say, okay, you have to be certain particle size and whatnot. But then I think it's still something in the works. What I would propose, though, is that there are already existing tests, and can we modify them for these microplastic analysis? That might be something easier to go for, because after all, it's something that a lot of bodies are already doing every day. And to a certain extent, when particles or whatever, that fiber is too thin, that's when this development would be needed, and that's when people would have to agree on, okay, is that level of detail required for, let's say, causing fish to get sick, or population of certain species to disappear.
Laura, do you have
Sounds
anything to
very
say?
standard.
Sorry, yeah.
I think I was just going to echo what I said earlier about being able to liken it to concentration, which then enables you to compare with, the existing regulations and compounds and things like, we have biotoxics regulations and things like that already, and being able to compare to existing things like that.
Yeah. Prof. Otani Sensei, do you have any feedback on this? Like what to expect in 5 years?
I basically agree with Dr. Barbara. Both technique is useful. And for example, in the case some particles are isolated, the spectroscopic method is very useful. But in the mixture samples, the thermal method is preferable, I think.
Yeah, I think you're all right. One thing is that we have insufficient data on this toxicological aspect. Okay, so I think in Singapore there are a couple of groups they are working on this, and you will listen to some of them on third day, because at least I remember one of the paper where they have actually ingested these microplastics and nanoplastics to the larvae, and they have seen the impact. Okay, so when it is ingested at high concentration, like 25 ppm and above, and you can see this the growth hindrance, and it's also dying. So that means you see the impact, like a acute toxic effect at very high concentration. But in reality, you don't see such a high concentration in the real world samples, and sometimes that will reduce the motivation. Okay, if the problem is only at 25 ppm and we only see few ppb, why we bother? So that's the kind of first feedback or thought may come to the people. But the further studies on that also reveal that there is a accumulation. Okay, so that means even if it is low concentration, you cannot ignore. So it will pop up at some point, so there are accumulation process, and particularly it is seen in stomach, So that means even the smaller concentration, what you think is not a problem today, can become a problem like tomorrow or years later. So that's where we see definitely this toxicological data will help even the other scientists as well as the policymakers to understand, because they also do not want to impose something that is not necessary. This is a combination of two things, and definitely one thing is that we need to have more data on the toxicology. To accomplish that, as an analytical scientist, what we can contribute is basically a good technique, and that can measure this at different matrices at different concentration level. And one of the point also I picked during today's presentation was the contamination, because in general, be it heavy metals or other microorganic pollutants, when you are trying to do ultra trace level, contamination is always a big challenge. And today, I think Dr. Barbara explained about that, and also I think there was another breakout session where it was mentioned as well, that they have come up really a kind of recommendation to have contamination-free lab to have a good kind of sample preparation. So how do you see this, the sample preparation and the contamination in both techniques? Okay, so we have to compare the challenges, because if somebody say I have a technique where I don't bother about contamination, then as a chemist or the daily chemists who want to analyze 100 samples, they want to have a robust and quick and dirty way of collecting data. So how do you see this challenge for the spectroscopy? I think we have listened to Dr. Barbara on the thermal analytical technique, but at least I want to hear from Martin. And how do you see the sample contamination as a challenge in spectroscopy?
It's something that can be avoided already. It's not the most straightforward process, but if you make sure that, well, we've already heard it in a talk earlier today, when you make sure that you're working in a clean environment, that you have air filtration, working on a hood at least, on a laminar flow, that prevents any kind of contamination from the outside when you're doing your preparation and your measurement, using glassware wherever you can, so avoid any kind of equipment that might contaminate the sample further, and store it in a glass Petri dish, of course, if you're transporting it when you're done it, when you're storing it. So this, I think if the lab is set up properly, I mean, our instrument is also small enough to just place it under a laminar flow so that the measurement is also not abstracted or is not something where the contamination can come in. This shouldn't be too big of a problem.
Okay. I think, just to consolidate today's discussion. So if I talk to anybody,
may I just add something here? I think
sure.
that is a good point, and it was said today often, but I strongly suggest to always do these blanks, because just a clean room and just clean conditions do not mandatory mean that there is no contamination. So
yes, definitely. I didn't mention this, but this is definitely very important to always measure blanks.
Yes, and the contamination even is in chemicals for the preparation. And it is really a hard work, because those chemicals are usually synthesized or produced at places where nobody cares about any plastic contamination. So they are stored in plastic bottles, or whatever is done. They have never thought to be tested for a microplastic contamination. Maybe for POPs or for really contaminants, defined contaminants, you get really graded chemicals, but you don't get them for microplastic. And we have had a lot of surprises when using chemicals, and even I think for pyrolysis you don't have any chance to differentiate. But even for spectroscopy, it is really, really necessary not to believe on your conditions, but really show and measure the procedural blanks.
Right.
Yep. And then may I also add that some of these particles are going to be pretty small. And then when there's enough of these, they will make a big clump of material, even in the blanks. So maybe it's good to check it step by step, and then have this, system and instrument with the higher sensitivity possible. That would be very helpful.
Sure. I think it was a very good and wonderful discussion. And as analytical scientists, we play an important role. So I think that's something we'll come to know from tomorrow's and day after tomorrow panel discussion. And at least our vision, at least the interest is not to end with this symposium. We just have to see how we can really put our hands together and really go together and share knowledge together. So that will allow us to advance on this, and whereby we have something concrete for the new people. Today somebody want to work, some countries they want to start research on microplastic, or they want to do something. There are general question, okay, which technique I should use, how to collect sample, how to prepare sample. There are you have 10 questions, always a questionnaire it comes. Thus, your front-end researchers, if we have some kind of recommendation, okay, and again, earlier we heard to different, we are not interested to say, okay, this is the best technique. There is nothing called best. So there's always a complementary, and it is based on what you want to do or what you want to accomplish. So it's just a combination of things. But it's important to disseminate the information, what every researcher get it, whereby the knowledge is reaching to the community, and that will allow them to do the next step rather than reinventing things what has been already understood. So that's something very key. So at least on day 3 we will try to do something on this and see what we can do with this forum, because now we have almost world-renowned people and also the technology developers, and at least we see what we can do and how we contribute to this problem. Anybody wants to add anything here? And we will open the floor for a question from audience. But we don't have much people because everybody is rounding in now. So, but anybody wants to say anything, please feel free to add something here. Yeah, Lo, you want to say something?
Yeah. So I think, like, with as I have discussed in my talk earlier, I think that having accurate identification of the microplastic fragment is very important. It's always good to start with good data, like to begin with, and then analyze the data with good software. I think that's good. I think that's the good combination that we have. But we got to start with good data, good spectra with good bench rate, so that we know exactly what we have. We don't want to have things that doesn't matter, like rough surfaces causing all the dispersion artifacts, and also the size shape dependence of the spectra. We just want a chemistry. And with the newer OPTIR technique, that's where I think we can get the chemistry that we want from the microplastics. But of course, I mean, this is just one technique. We have like IR plus Raman, but then it's always good to have another check with the thermal analysis. So, I mean, it's more like, it's not like, either you or me, but it's more like we are working together, to get the confirmation that we want and that we need for scientific research on these microplastics. But again, we got to start with good data. I mean, we the best data we can, so that we can, prepare a good meal. And a good data. Good report, of course. That's what I mean to say. But,
Yeah.
And I think Raj can.
Yeah. Anybody want to say anything more here? So before we really conclude the session, Dr. Othani, you have anything to say?
No, thank you very much.
Okay. Barbara? Okay, good. Yeah, done enough, right? Laura, anything you want to add? So what we have
No,
discussed?
not from me. Thanks. That was it's been a really interesting morning for me. Thank you, everyone.
Okay, Bentik, anything more from your side?
Oh, no, but I think we have one question from the audience, if I'm correct.
Is it? Okay, I was looking there. I did not see any.
Are standard reference materials available for microplastics?
Okay, that's a good question. And yeah, I think I have some answer, but it's not the concrete answer, because there are some standard materials available. It's not really certified as a reference material yet, but it would be possible, that would be possible. Again, that's why this forum can really come forward to do that. Because if I have a material where I know the composition, then I give it to Mike, I give it to Martin. We put our data together, and that really become a reference material for everybody. So that will allow us. So yeah, the answer is we have half answered for now.
Yeah, that could be a good start. I think having reference. I mean, we already have standard material for, let's say, microplastics for cytometry, as well as for let's say the potential measurements, these NIST traceable particles. But then, of course, these are spherical shapes.
Yeah, exactly.
So these are not real. They don't represent the size and shapes and the irregularity. I mean, that's the most important part, the irregularity of the microplastic fragments that we are actually that I see almost very frequently from our customer samples. They are different shapes, films or chunks or whatever it is. So I think I've seen papers where people would actually manufacture their own, they use a filer just to get different sizes and shapes. I mean, it's a good approach, but I think having a NIST, some type of NIST traceable standard would be quite helpful in helping us. But of course, because it's quite variable. I think the control in manufacturing such type of granularity would be quite challenging, I think. But still would be nice, I think.
Yeah. Before I wrap up, I just have one question to the spectroscopic side, because I'm also part of this thermal analytical group. So we always believe the importance of the mass quantification. Okay. So as a spectroscopic technique, sometimes we drive through the particle counts and particle identification. But how do you see if I have to really know the mass concentration of the particles in a matrix? Is it feasible, right? It's feasible. So can you share, like Matan and Mike can share some thoughts on this?
If you want, I could share some thoughts.
Please, yeah.
Regarding, for example, hyperspectral imaging techniques, there has been different research papers published for computing mass content from spectroscopic data. So, for example, by detecting the particles and assuming a third dimension, and then a very simple particle model is applied to, by using the density, you can then compute the mass. However, one really has to be careful regarding these models because the third dimension is just an assumption. And I think there is still work to be done in order to be really reliably measure this. And I'm not sure whether this is really possible. It's an interesting research question, but one has to be critical regarding these kinds of data.
Yeah, when I can chip in here, Benedict is basically correct. So we always have 2D data and have to estimate with some kind of model the third dimension. And I think there's some room for improvement still. We can still become better because at the moment what I've seen, the mass estimation differs quite a lot from what mass spectrometry in the end delivers. But I think we can get closer if we adjust this model better.
Maybe I can comment on that as well, because we have already done those comparisons. And I think, as already told, that is really a challenge to compare those data, because, and that is something which we haven't pointed out today, but it's very important that before you compare spectroscopical data to mass spectrometry data, you really have to do another harmonization step to speak about the same polymers in the first step. And as I told you today, this cluster problem just in between mass spectrometry is one side, the spectroscopical persons clustering as well, because some polymers are really hard to distinguish. So if you are going to compare these data, another harmonization has to be performed to talk about almost the same groups. And even then, it is really sample dependent if you got a more or less reliable approach. So what we found out that the general trend in transects or whatever is very comparable between both methods. So that is quite reliable, but the masses themselves are very, very tricky. And I think that is something one should think about, because we hardly any know about these particles in a small size and chemical change, etc., in density, whatever. So it is very, very difficult. It is a broad assumption and comparison, but another harmonization step is hardly wanted before you do anything else.
Definitely, yeah, it's a good point, because at least personally, I will explore the possibility to have some kind of intercomparison where we come up with some common homogenized material that can be analyzed by different technique. At least we can have two objectives. One is to identify the polymer present in this material. Then, if possible, go to the next level of quantification. Okay, with every technique, then we can really use this as a base for us to.
But it's an artificial sample. And I think that's the problem. I think what we are needing are real environmental samples, and then the standardization of those environmental samples, once with spectroscopical techniques and once with thermal analytical techniques, and then use these environmental samples as a quality standard to distribute between the labs, because then we have real conditions and not always artificial.
You're absolutely correct. But if you look at some of our proficiency test exercise or the interlaboratory exercise, because real world samples, sometimes the concentrations are too low for intercomparison. Okay, again, because if I have to give you a seawater, okay, for some kind of analysis, I have to give a barrel of water. And this is something difficult to really do in a large group of people. So that's why even if you look at some of the proficiency test exercise, they try to have a matrix match standard. So that means they have to take the matrix, then they do some spiking, with the hormone. But yeah, definitely this is the next level we should see. But to start somewhere, it's something that we can really think of a material that can be at least identified by all these techniques, and we get some kind of good agreement that is increasing the confidence of everybody, and also the users.
Yeah, totally agree, Karthik. I think as well as to Barbara, I think the complexity of the real microplastic, even just the polymer itself, can have multiple species. That would really throw up, all the analysis that we have, makes it very complex and also difficult to estimate the density. I think that, but still having, I think for spectroscopists like us, we always try to figure out, okay, what is exactly in there? I think that is one part of the equation. And then, for us, it's more like a non-destructive technique. I mean, I think this is the helpful part. When we analyze our sample, it's not destructive. So at least what we can do is that, let's say we have done our analysis on the spectroscopy side, then we can use the same sample and analyze it in the mass spectroscopy and thermal analysis side. At least we have some way to build some sort of statistics, So at least, I think that's the part where I think spectroscopy can help preserving the sample. It's the order of the analysis that could potentially help. Now, of course, it's still an estimation. Mass is not an easy answer. Not an easy question to answer, really. How much, how many milligrams of this stuff do I have? But Martin said, it's a two-dimensional problem. We always get the 2D image. How can we possibly get a volume? And then, of course, if we know the identity of this copolymer of some sort of multiple component, then we can get the estimated size. But still, it's going to be an estimate. There's going to be an error bar to it. We can't just say, oh, it's exactly 1,000 milligrams. I don't think so. But, at least having some sort of correlation between spectroscopy, we have good software in analyzing the shape and particle size, and then the identity of the chemistry, that is, the real actual chemistry that is in that particle, and then how well is that going to compare with, other techniques. I think, having some sort of, agreement between those two would be quite, I think it's a good case. And again, it has to start with good data.
It's exactly the right way to do the successive analysis. I think that is the best. And if you have both techniques, then you have a solid database and a really good starting point for any argumentation.
Mm-hmm. Good starting spectra. It's important.
Okay, I think we can end the session with this. I think we have almost like a 1 hour discussion. And first, from organizer side, I have to apologize because we had some hiccup during this breakout session, because of which we got delayed, and all presentation also a delayed bit here and there. So overall, we were running late by 1 hour, but we had the energy to run 1 hour discussion, which is really great. And we have this recording, so at least after the symposium, we'll try to put these things together and see what we can take forward. And our discussion should continue and see, and once in a while we should consider meeting up for such interaction like a symposium, so that can be rotated also. All this we will explore because it's the first edition for our region. And so when we say something for Southeast Asia, doesn't mean that we only have to have researchers from here. We need European researchers to come to contribute, and we just have to do this way. And this is the first edition, and any learnings and things that we get out of this, we will try to continue, to have our knowledge sharing and consortium, so that we can work together, develop things together that will solve some of the society challenges. Okay, so with that, I will end this the panel discussion. I thank you all for your time and contribution. So today was an excellent day with a lot of information, and most of that thing already I got a lot of feedback from the participants. It was wonderful, and everybody felt the time is not enough. So that's always the case. Yeah, but still we were able to entertain our audience almost for 5-6 hours. Only now people were a bit tired. I think until the panel discussion side, we had 70 people always on live, out of 100 over people. So it was a good day, and we will continue our interaction. So tomorrow, most of the talks are related to the real world problem. Some people have done some analysis, collected sample from different environment. Some have have done some treatment. So I think whatever we have said is missing. Maybe some answers tomorrow. Then the third day, they will be talking about some of the toxicological aspect. Then all these three together will give some kind of direction for our future research. Okay, so with that, I thank you all. Then we will close this session and have a nice evening. And also, like I said, good night for some of the people here. So it's already okay. Thank you very much, and let's all meet again tomorrow. Okay. I know some of you are very busy, but please try to join whenever is possible, so that you are not out of track.
All right, thank you so much.
Thank you very much.
Thank you very much.
Thank you,
Bye-bye.
everybody.
Thank you.
Nice meeting you
Bye-bye.
all. Thank you to the audience. Bye-bye.
Bye.